Okay, now we are moving on to the next segment of panel discussion, question and answer. I'm the moderator. I'm Joachim Loo from the School of Material Science and Engineering. We have heard a whole spectrum of topics this afternoon. I'm ranging from experimental results to theoretical experiments to crystallography, and the list goes on. Of course, all within the area and the field of solar energy. So since after hearing all the talks on energy, I think we should have an energetic and robust discussion with our guests over here. I'd like to start off with the very first question for Professor James Barber. Professor Barber, you have been an eminent scientist working in the area of natural photosynthesis, and then you have seen how science has evolved to today. You're also working on synthetic photosynthesis. What are your thoughts on this, and how do you foresee the future?
Well, I'm very lucky that excuse me, I've got a bit of a frog in my throat. I'm very lucky that I decided when I was a young man that I wanted to work in photosynthesis, which after all is the great photochemical process of this planet. The intersection of light with matter and its conversion into an energy source. And so I saw it as the big picture, the big photochemical process of the planet. And that has been my drive. It's not always been easy, I've got to say, because in a university, it's quite difficult sometimes to get continuity of funding. But nevertheless, I didn't lose my focus. Whenever I could work on photosystem 2, the water-splitting enzyme, I concentrated on it whenever I could. We did, over the years, and some other people, including my neighbor here, worked on the biochemistry and the molecular biology. I didn't have time to talk about all that. But in the end of the day, and we determined the structure of all those proteins. I haven't talked about any of that, and that was all marvelous. But in the end of the day, we want to understand how the molecular machine works. And we've just heard a wonderful lecture on the ATP synthase. And this formation of dioxygen, all the oxygen in the atmosphere is derived from that process. This planet would not have oxygen on the in its atmosphere if it wasn't for the photosynthetic water-slitting process. So it is, and it's the very first chemical conversion reaction of the planet. Everything else thermodynamically is downhill from there on. So all the other energy processes that occur in us, for example, are all a result of that energy storage by splitting water. That was my drive, that it was such an important reaction on the planet and in nature. And I focused on that. I got to tell you that at the time when I started working on this, I never believed we would ever get a structure, or I could give a lecture today of the detail of the possible chemistry of them, with a certain amount of certainty now about how the enzyme works. It seemed impossible. But I often say in a situation like this, what seems impossible becomes possible as long as you focus your attention and put an effort into it. And that's what I was lucky enough to do, and that kept me going. Now, the next lucky break, in a sense, is our concern about the future of this planet and about how we're going to continue to use fossil fuels as an energy source. As I said, 85% of our energy, or nearly 90%, comes from fossil fuels. We all know we cannot continue. Oh, I should say there is a lot of fossil fuels on our planet, because if there's 21% of oxygen in the atmosphere and only 0.04% of CO2, then, and all that oxygen's come from photosynthesis, then you know that there's been a tremendous amount of reduced carbon on our planet. That means there's probably unlimited amount of fossil fuels. People don't often say, so we'll run out of oil, or we'll run out of coal, or out of gas. It will not happen. There is so much there. It may not be possible to get it all, but it's there. And in principle, if we burn all the fossil fuels, we would have no oxygen in the atmosphere, and we would have an atmosphere with CO2. So it's that challenge. It's been set up by the Paris Accord of 2015, that we must try to, as a global society, we must try to reduce the CO2 levels in the atmosphere, or the release of CO2 into the atmosphere, because of the tremendous effects it may have on, well, it would have ultimately on the climate of the planet, and disruption of human society. So I've been drawn into that and say, okay, if the leaf can do it, we can do it. but we've got to do it better than the leaf. We've got to have catalysts that can split water and make fuel available in a continuous way, which is clean and non-polluting and doesn't rely on fossil fuels. We have to do that. Otherwise, and I cannot see any other solution but to use sunlight, nuclear power, which is available to us in enormous amounts, as I said in my lecture. So I believe we have to work very hard at this goal, and I'm very honoured, and I thank NTU and my colleague Professor Anderson for allowing me to work with very talented people here at NTU, and we have achieved a lot. I would like to tell you how many papers we've published, but we've achieved a lot. We have not. We are only a part of a journey to get a new technology to give to the human society the opportunity to continue on. And after all, we're unlike any other species. We are able to dictate our futures. If you're a bacterium on a petri dish, you grow and grow and grow, and then you die because you've run out of resources. And we're going to do that if we don't use our brains. There's already 7.5 billion people on the planet. We cannot continue to grow our population and the expectations of the population without having new knowledge to deal with the problems that brings.
Has this been recorded? We could send it to the White House in Washington.
I'm very happy to. I'm very happy to speak to the White House.
Thank you very much, Professor Barber. Questions? Yes, the gentleman.
Great. Thank you all for a great symposium. And just to say, I am from the U.S., and this is why I came to Singapore. So my first question is for Professor Barber. Mainly, I'm not sure if everyone knows this in the field or not, but is there only one type of photosystem 1 and 2? Basically, do all plants use this one type of system? And if that is not the case, what are some other examples?
I have no reason to think there's any variation on the theme, that this catalytic center that I described in my lecture is unique. It's a unique chemical system, not found in any other enzyme, and I believe that every oxygen-producing photosynthetic organism has exactly the same structure. And we know from the proteins that they're highly conserved. One of the main proteins that binds the manganese cluster is the most conserved protein in nature. And you can see it all the way through from simple bacteria, cyanobacteria, right the way through to higher plants.
So could you see any other mechanisms that would work in nature? And if so, why do they not exist?
There are essentially two mechanisms for forming dioxygen from the chemistry point of view, and that would be radical coupling, OO coupling, and also the nucleophilic attack mechanism. Now, the radical coupling mechanism has been suggested. It was suggested, significantly suggested, before we had a structure. But the structure lends itself very much to the nucleophilic attack, by the way that you have a bimetal center, comparison with other enzymes, not only with carbon monoxide dehydrogenase, I explained, but also actually with cytochrome oxidase, which does a reverse reaction. So I'm pretty convinced that that is the reaction. But there are others who are not, and there is at the moment, there's quite a controversy going on, and I didn't enter into it, which involves a mechanism in which the substrate water molecule breaks up the cubane and enters in, and you get actually an oxo radical oxo mechanism. And I don't feel comfortable with this mechanism because I believe in a surface mechanism because I know that cubane is extremely stable. I told you it can be synthesized. It seems ridiculous in chemical terms to break it open and reform it again. And the other thing that's very important about photosystem 2 water splitting is that the overpotential for each step is very small. That is, it's just got about enough energy to drive it. There's about 4 volts in total, 4 photons at 1 volt. And if you start having large conformational changes, opening up this cubane structure and reforming it, This is additional energy that would have to be required and not compatible with such a low overpotential.
Okay, thank you.
But it is very controversial at the moment.
Well, can you check that with isotopic mapping or anything like this?
Pardon?
Can you test if it goes into the cubate with isotopic mapping of oxygen, for example? Like to see if
the isotopic experiments have been done. There have been many of them. Support really a surface binding of water molecules at the very beginning of the S-state cycle. The mechanism that's been advocated by a good friend of ours, Per Sigman in Stockholm, has a substrate water molecule binding in the cubane at the S3 state. There's no evidence for that based on isotopic exchange. But you know, nothing is black and white. And, and at the moment, there have been two very fancy experiments done using XFEL, X-ray free electron laser diffraction. And this is time-resolved X-ray diffraction. And they've come to the opposite conclusions at the moment. They're both published in Nature in the last month or two. And so the jury is out. The other problem is that the reactant, this is a unique enzyme because the reactant is water and the solvent is water. So that what other enzyme do you have that? And the reactants are oxygen, O rather, HO and H2O. Those are the reactants. And the difference between them is 1 or 2 protons. We could never detect that difference by X-ray crystallography or by the XFEL. You may detect some other larger movements, but you will not detect the proton. It's virtually impossible. You'd have to have resolutions less than an angstrom. It is very, very unlikely. That's not to say there aren't other techniques that will come into play at appropriate time. Proton diffraction, for example, neutron diffraction, excuse me. But at the moment, I would say we don't have the techniques to really sort it right out. But we can eliminate certain mechanisms, and I think that, yeah.
Thanks.
Can I ask, are the free electron data different, or are the interpretations different?
Well, this is a big question. The papers are different. The interpretations are different. The data needs to be looked at independently to compare the data. And in fact, this is something I'm doing with my colleague James Murray at Imperial College, because when you have a paper of that complexity, you have to have the primary data to go back and test what the conclusions are. That primary data is not available when the papers are submitted to Nature in this case. So, yeah.
Thank you very much.
So,
Yes.
I'd like to throw a grenade of a different type.
Oh, good. Not to
Okay.
me.
No, not you're welcome to answer, but I think in the interest of the flow, short answers are useful. So, photosynthesis is a terrific way of producing energy, and I wonder if any of you who come from, let's say, the non-biological domain, remember where the idea for the battery came from. So Volta, who put that idea together, what he did to come to that was he looked at the Amazonian eel, the Electrophorus electricus, which forms a set of cells to produce 600-volt shocks. So I asked the group, have you thought of other ways of using photosynthesis to charge batteries using synthetic biology to actually make plants that mimic the eel by placing... So I'll give this one to Bertel because he's creative and he needs a new career soon, right?
That's right.
I mean, why not use synthetic biology to put sodium channels and sodium-potassium ATPase, which charges the penny pile of the electric eel, use photoactivatable channels, okay, to kick it off, and charge it with a derivative of photosynthesis.
I memean, we're doing some synthetic biology here with the cyanobacteria, and we're trying to get one gene into it to do hyaluronic acid, and that has taken 2 years. So I think nature has gone on here now for 3 billion years, so maybe we should utilize that. But, I mean, as an intellectual discussion, I think it's very challenging.
Any takers?
Like I said, I'm not going to because I've spoken too much. I think that in the end of the day, a technology that we need on a vast scale has to be robust. It has to be efficient. And I don't believe going through a biological route would be the way to go. Photosynthesis.
Trees use a biological route, right?
But
Trees.
they are, but they're very inefficient. They're 0.01% or 0.1%, excuse me, because they have to grow and the leaves fall off in the winter and maintain themselves. So we need something that's much more efficient than photosynthesis. The leaf can do it, but we have to do it better.
Professor Grezel?
Yeah, I think we all can learn from natural systems, and so the eel that's a very, very interesting system, because I mean, to get these high voltages without breakdown, that's amazing. So I think your thought is well conceived. You just have to see whether this can be now implemented in a more robust way than organic matter. But the fundamental is amazing that just by, did you say just by moving ions around, there must be some electronic charge separation as well? How the eel.
The cells are big. They're thin as vellum. One side of the cell has sodium pumps.
Yeah.
Charged by ATP.
Okay.
The other side has a voltage-dependent channel, like your neurons use,
Mhm.
for you to think. And
I
talk.
see. Okay. It's all
And
done
it
by.
displaces them across
Okay.
about a 20-micron space filled
Okay.
with
Yeah.
keratin-like filaments
Okay.
and water.
Okay. It's just like a capacitive charging.
Professor Barber?
Yeah, I think the idea really to look what nature has developed in all these hundreds of millions of years is always good. You know, we have surprising examples where we have learned from nature, and now it is mainstream technology. In terms of what we are talking about, not now the storage, but the power production from sunlight, there was a very strong effort to grow power plants and convert them to fuels. Actually, it's still going on in Brazil and so on. But of course, this is a very inefficient way because of the inefficiency of the photosynthesis process, as we discussed. So if we use the same area of the land to cover it with solar cells, then we get much more power than we use power plants. There's one area which I think the word is still out, and this is to grow algae. Because algae we can grow in ocean, in water, in areas which are not used and which cannot easily be used for other ways of collecting the solar light. So maybe the algae process might indeed turn out to be a very powerful way to create biomass from the sun and use it for power production. But it's too early to say if at the end of the day this will. But definitely from nature we can learn a lot. For instance, I remember in our institute we studied what is the best way to make a photothermal module. You know, how to create the water flow so that it's most efficient with the least amount of water tubing, getting most efficient heat exchange. And the structure at the end is just the structure of a leaf. You know, therefore we call it a bionic system, because nature has really optimized the flow structures inside a leaf, and that is exactly where we should learn. So wherever we can learn from nature, let's go for it.
Thank you very much. The next question. The gentleman. Yeah.
Thanks for the talk. I have a question a bit more on the philosophical side. That we have learned a lot from the natural systems, and it was constantly being said that the efficiencies are not very good, although nature does it very well, but the efficiencies are not very good. We can make photoelectrical chemical cells the best we could do, but still we are lagging in terms of catalyst, in terms of the turnover numbers. So is it the best solution lies somewhere in between, or the nature has already chosen the best way, because that's how the nature has evolved in the billions of years? So is it the nature has already chosen the best option, or is it the option lies somewhere in between and we are going at the right track?
Who's the question to?
So the question is that
It's to anyone.
No, I know the question. What is fantastic are like these ruthenium complexes, for example. They're not driven by light; they're driven by adding an oxidant. But they have turnover rates much, much faster than photosystem 2. So we can get catalysts that have very high turnover rates. But that is very important. If we're going to use light energy and we're going to have a lot of energy, we need catalysts that turn over very rapidly. Photosystem 2 does turn over very rapidly when it's isolated. But of course, going back to what we were talking about before, if it's then linked into much slower processes, carbon fixation and leaf growth and so on and so forth, of course, that affects the efficiency. But when you take parts of the photosynthetic process, like light absorption, excitation transfer, we heard about charge separation, and the chemistry of water splitting, in isolation, they're very efficient. But when you put it all in as a long chain, the whole thing has this low efficiency. Especially we have to bring in the environment and the access to water and winter and summer and so on and so forth. All that brings the efficiency down. So an artificial system needs to learn from the natural process, but pick the bit that is efficient. That is light capture and initial energy conversion.
And I think the systems, the plant are quite successful, so even if it's low efficiency, it's efficient enough, right? And it's interesting, Jim and I wrote an article some years ago about light can be bad and good for photosynthesis. So this marvelous machine that Jim told about needs to be repaired all the time because the chemistry of splitting water is quite dangerous, you could say, with light. So I think, was it 10% of all the ATP produced in the plant cell is used to repair the machine in a way, So it's tuned in a different way, and if you have to take it out of the cell, you need to, you have to make it stable, but you can make it much efficient.
In a photovoltaic, as you increase the light intensity, the current goes up and up and up. In photosynthesis, as you increase the light intensity, it will go up and then it will saturate because of all the other reactions that are going on.
So in the interest of time, let's have one last question. Professor Subodh.
Could I just extend what Sudhanshu said? Can we look at genetic modification of photosynthesis? Is that a possibility? Can we get 10-20% efficient photosynthesis going?
I think you can, but not in these primary reactions as we talked about. Jim just said that one of these protein is one of the most conserved in nature. So if it could be variable, it would not be that way. But what you can do in the plant system, of course, is to make it more efficient through some regulatory processes and so on. It can adapt more and so on, but not for the artificial systems.
Thank you very much. So before we end off this panel discussion, I'd like to just reiterate the quotation from Professor James Barber. If the leaf can do it, so can we. Thank you very much.
I'd like to say thank you very much for coming. It's a real honor to be here. And thank you for coming and sharing this wonderful symposium with me, which I'm very grateful to the speakers who've made the effort to be here and give wonderful talks.
Thank you.
Thank you so much. Let us thank the speakers and Professor Barber one more time.